initially I was looking at protein interactions and how they play a role in regulating specific cellular pathways. But what I became more interested in over time was the role of lipids, okay? And in particular, how lipid microdomains at the plasma membrane were involved in regulating these protein complexes. And I guess from there, we became more interested in actually the modification of those proteins. We know that mutations in the enzymes that regulate lipidation are linked to intellectual disability, to epilepsy, to cancer, to diabetes. So what we hope to do is to understand at the molecular and the cellular level how those defects in lipidation are actually causing those important disorders and diseases. So on top of that, so that's I guess is giving us insight into clinical conditions, but also what we hope to do is to validate a number of these enzymes as drug targets, and also in the future hopefully to kind of develop kind of molecules that can modulate the activity of those enzymes, which kind of 5, 10, 15 years down the line may find kind of utility in a therapeutic sense. So the technology contained within the Strathclyde Centre of Biomolecular Sciences will enable our research, I think, at many levels. Okay, so you've got mass spectrometry facilities, which are very important for our research. So they will help us, for example, to map out the substrate networks of individual lipidation enzymes. The metabolomic facilities will allow us to understand how metabolism is disrupted in these different disease states that are linked to lipidation. We've also got new super resolution imaging facilities, such as the STED microscopy. So that is going to be very important to us in understanding how lipidation regulates the localization and the micro-localisation of proteins inside the cell.